And our last speaker today is Dr. Mohamady El-Gaby. Mohamady is a postdoctoral neuroscientist at the Nuffield Department of Clinical Neuroscience at the University of Oxford. And he currently works in the lab of Tim Behrens, but will be presenting work from his previous postdoc with David Dupré. And today he's going to talk about a neural coactivity code for dynamic memory.
Thank you very much, Elaine, for this kind introduction. I'm going to be talking a little bit about the role of neural coactivity in encoding information and in supporting memory. And the sort of central question that we're interested in is what features of neural activity encode information. Now, consider this sort of toy example here, where you have a set of 6 neurons that fire differently to different stimuli, X and Y. So neurons 1, 2, and 3 need to fire more for stimulus Y, whereas 4, 5, and 6 fire more for stimulus X. So this is your standard sort of rate code that is sort of just throughout multiple species and systems. But then think of this second example here on the right, where now there's nothing about the single order or first order features of these neurons that encodes information about X and Y. However, if you look closely, you can see that neurons 1, 2, and 3 are highly coactive within a short time window in response to Y, whereas 4, 5, and 6 seem to be highly coactive or highly synchronous in response to stimulus X. So here the information about the stimuli is present as a sort of emergent feature or an emergent property of short timescale coactivity. So I'm going to be referring to this type of code as an emergent coactivity code. And this kind of this type of code has been sporadically reported in the literature in certain species and in certain systems, but the cognitive function that it might be playing has not been explored. And so our sort of sub-question that we're interested in is what are these emergent short timescale coactivity codes useful for? And we hypothesized that perhaps in situations where animals must form dynamic memories, that is, memories that are rapidly formed and flexibly retrieved, you might need to form or encode this information using short timescale coactivity, given the speed of both stabilizing these coactivity codes and also retrieving them. And sort of to further clarify what I mean by dynamic memories, let's just jump straight into the task that we're using. I designed a task for mice in which mice have to learn two new behavioral contingencies every day. And the contingencies look something like this. So when a tone comes on in the presence of one set of LED displays, the animal needs to go to one of 2 dispensers, in this case dispenser 1, in order to get sucrose, and needs to avoid an identical looking dispenser in a different location, which delivers quinine, which is quite a bitter substance. And then in the presence of the same tone, but when a different set of LED displays is illuminated, now the contingency is reversed. So dispenser 2 is now the good one, the one that delivers sucrose, and dispenser 1 delivers quinine. So the animal has to do to approach a different dispenser. And the animal learns a new example of this every day, and we can show that the animal starts off naive. On a given day, the structure is as follows. First of all, animals will explore the learning enclosure in the absence of any tones or sucrose or quinine, but in the presence of the LED displays, and also explore a control enclosure. And then it receives 2 learning sessions in each contingency. And then finally, after the last learning session, an hour later, the animal is tested in a memory probe test in extinction. So the tone is delivered, but no sucrose or quinine are given. And crucially, in that probe session, while the animal is in the same enclosure, we pseudo-randomly switch between the 2 LED displays. So the animals have to quickly update their behavior to be in line with the contingency associated with the LED displays. And so this satisfies both of the criteria that we've set for a dynamic memory task. Animals have to learn it rapidly every day within a couple of hours, a few hours, but also they have to retrieve it dynamically because we shift between these LED displays. And what we see is the animals indeed do remember, or retain a memory of this contingency discrimination into the protest. And this is sort of a pictorial representation of roughly what they're learning in this task. But this is nice because it gives us a dynamic memory paradigm in which we can now test hypotheses about neural mechanisms. And for various reasons, we thought that the dorsal hippocampus might be important. So we recorded from dorsal CA1 as the mice performed this task. And we could record from multiple individual neurons. Here I'm going to talk about data from principal or putative principal neurons. And crucially, in order to get at coactivity, we can detect patterns of coactivity using a method that relies on independent component analysis to isolate these coactivity patterns displayed here in these stem plots, in which we use these sort of weight vectors to display the weight of each neuron that's been recorded in the population to a given coactivity pattern. So this is one coactivity pattern, this is another, and so on. And this allows us to do multiple things. It allows us to track the strength of these coactivity patterns across time with a high precision, as you can see in this blue plot here. It allows us to also see which patterns are detected in which sessions, but also we can isolate the highly contributing neurons, so the neurons highlighted in color here in blue or here in red, and see and look at the properties of these neurons. So one of the first things that we did was to see if there are patterns that are present in one contingency and not the other. And so if you look at this similarity matrix here, you can see that highlighted in blue is a pattern that is present in both contingencies, which we're going to refer to as contingency invariant, whereas highlighted in orange is a pattern that is only present in one of the two contingencies. And so we can isolate these two types of patterns, and sort of their similarity profiles are shown here on the right. So we do see some short timescale coactivity patterns. Crucially, all of these coactivity patterns that I've talked about so far have been detected in a short time window, in a 25 millisecond time window, which roughly corresponds to the membrane time constant of these principal pyramidal neurons. And so we can see some contingency discrimination in these coactivity patterns in CA1. Now, the next question that we asked was is whether any of these patterns or whether these contingency discriminating patterns are indeed showing an emergent coding that is a function of their coactivity. In other words, are the properties of the individual neurons somehow also correlated with contingency? And so as a sanity check, we first of all just check that the sort of Pearson correlations between neurons within a given coactivity pattern are higher in the preferred contingency, and that is indeed the case. But now we were interested in this first order properties. So are these neurons also disambiguating contingency by their rate or other first order property? And first of all, we looked at their firing rate across contingencies, and that's not different. We also looked at their firing rate at the correct and incorrect location, so the sucrose versus quinine, and that's not different either. There's no relationship between their firing rate and distance from the goal. But also, even when we look at the single neuron temporal properties, so for example, when we look at the interspike intervals distribution across contingencies, that's not different either. And the temporal tuning to the actual trial stages themselves, so the onset of the tone, the onset of drop delivery is not different across contingencies. And even when we look at their tuning to theta cycles, so the theta phase coherence of these member neurons is not different across contingencies. So basically, every single neuron measure that we've looked at so far shows that there is no coding of contingency at a single neuron level. It seems like this is an emergent coactivity code for behavioral contingencies. And so this looks something like the schematic that I showed at the beginning, but now instead of sort of raw stimuli, we're looking at something a bit more abstract, sort of these contingencies. So we then asked whether this contingency discrimination somehow helps the animal perform in a task or has any relationship with task performance. And to do this, we simply track the expression strength of these coactivity patterns during exploration and learning. And what we see is that contingency invariant patterns are already increasing in strength during exploration, whereas contingency discriminating patterns seem to only robustly increase during learning. And a similar pattern is seen if you look at the reactivation of these patterns in sharp-wave ripples during sleep. So the sleep sessions either side of exploration, you see a reactivation of contingency invariant patterns, but not contingency discriminating ones. But the contingency discriminating patterns seem to be reactivated selectively in the sleep sessions after learning. So together, this data from both the online behavior and the offline reactivation suggests that contingency discriminating coactivity develops with learning. We can also relate it more directly to performance in the probe, and we see that contingency discriminating coactivity is stronger before correct compared to incorrect trials. The same is not true for contingency invariant patterns. And interestingly, if we look at longer timescale coactivity patterns that are contingency discriminating, while we can see contingency discrimination at these longer timescales, these patterns are not reinstated in a probe in a manner that predicts performance, suggesting that this is a particular feature of these short timescale coactivity patterns. And then finally, what we're interested in is to see whether these contingency discriminating coactivity patterns have any causal relationship with the actual behavioral performance of the animals. And so in order to do this, we silenced inputs to the CA1 coming from the left, CA3, which previous work has shown that these inputs are highly plastic. And we reasoned that if we silence them during the learning phase, we might disrupt contingency discriminating coactivity patterns from developing. And that is, sort of long story short, that is exactly what we see, despite so basic features of coactivity not being affected. And so we see this by looking at this using this ICA method, but we see this also using a different method that relies on pairwise correlations and Bayesian classification. And so we can get a mechanistic handle at these contingency discriminating coactivity patterns and see whether behavior is also impaired. And bearing in mind that we're silencing only during learning, but what we see is that during memory performance in the probe, the animals basically go to chance when you get rid of these patterns, suggesting that they may be necessary for this memory performance. If you silence the control pathway coming from the right, you see that you don't impair performance. And then finally, if you silence the same pathway, but in a one contingency learning task, you don't impair performance. So all of this together, I realize the time is up, but all of this together, I'll come back to this summary slide, but all of this together suggests that these contingency discriminating coactivity patterns are necessary for dynamic memory performance. And in the end, I would like to sort of thank all of the Gray Lab and especially people who are involved in the study, including David, Oli, and Vito. Thank you all for listening.
Thank you so much. That's really impressive work. I hope I'm unfortunately a bit out of my depth, so I hope there'll be some questions from the audience. But maybe could you talk a bit more about the mechanism of that blocks the coactivity patterns? Sure.
So the silencing of left CA3 inputs. So we previously, both myself and others in the Paulson lab showed that these inputs coming from the left CA3 show robust long-term potentiation, long-term depression. And so we reasoned, because these patterns seem to develop during learning, we reasoned that if we interfere with inputs that seem to be plastic, and also inputs that seem to be important for certain types of long-term memory, we might selectively interfere with them, and that's exactly what we find. These patterns are so this is this. Oops. This is this slide here, where we show that the distribution between contingency similarity of the independent components, or the coactivity patterns, is shifted in the light-on condition, shifted towards contingency invariance. So suggesting that we have indeed disrupted these contingency discriminating patterns.
Fantastic. Thank you. So we have a question here from Daniel. It says, coactivity coding sounds super interesting. Could you briefly explain the difference to phase coding?
Right, sure. So so phase coding would suggest that there's a something about the single neuron properties. So something about the locking of the spikes of a single neuron to a phase of some oscillation that tells you what stimulus is on. So let's say you've got an oscillation at the peak. If you fire at the peak, then stimulus X is on. If you fire at the trough, then stimulus Y is on. So that would be phase coding, and it's a single neuron code. Although you can imagine that it could be a coactivity code too. But the key difference is that a phase code doesn't have to be an emergent code. It doesn't have to be a property of multiple neurons. It can be a property of just one neuron. Whereas coactivity code is the information is only present if you look at the combined activity of multiple neurons.
We might try and squeeze in one more question from Nick Robinson. What happens if you repeat the analysis with 120 millisecond time bin? Do you see this occurrence of these discriminating events, particularly at a certain theta phase?
Right. So I think there's two questions there. So we do 120 millisecond. If we do longer bins, you can still detect contingency discriminating patterns. Although, so when I try 1 second, these patterns are not reinstated in a way that predicts performance. I haven't looked at sort of everything in between 25 and 1,000. The other question was about, sorry, was it about the phase of?
Yeah, we may not have time to answer it, but do you see the occurrence of these events at a certain theta phase?
Right. So there seems to be a slight bias, but not a statistically significant one, towards the members of these contingency discriminating patterns happening slightly earlier in the theta cycle, or firing slightly earlier in the theta cycle than the invariant ones. But it's a very small bias.
Well, thank you so much. That was a really interesting talk, and thank you to all of our panelists. I think we had a really good session.